it's now a privilege for me to introduce Professor Jay Tischfield of the Department of Genetics on the Bush campus, School of Arts and Sciences, and the originator of the Rutgers University Cell and DNA Repository. And Jay is going to tell us how Rutgers played a decisive role in combating the COVID-19 pandemic. Please go ahead, Jay. Thank you.
Am I on mute? Very good. Can you
Audible and audible.
Can you hear me? Can you see my screen?
We can see your screen, yeah.
Okay, wonderful. Thank you very much, Stephen. And thank you for the invitation to speak before this group. I'm honored to be before.
Jay, I'm sorry, you're a little static. Can you get closer to your microphone, please?
I'm going to switch off to a wired mic. I wasn't prepared to be the third speaker. I'll be right there.
Thank you.
Okay.
So for those of you who don't know, the test that Dr. Tischfield is going to talk about is even known to our president, to President Trump.
Can you hear me now?
Infinitely better, Jay. Thank you.
Ah, okay. Well, the marvels of wires versus Bluetooth. In any event, thank you again. I am honored to be in a group of people who know so much about coronaviruses. In fact, I'm not sure I was really terribly familiar with coronaviruses merely 6 months ago. I am not a virologist, and I've never been involved up until recently in diagnostic testing, but situations demand action, and it was kind of obvious in January that we needed a diagnostic test for SARS-CoV-2, and that the test that was made available initially didn't work because of issues with the PCR primers. So we also reasoned that if there were to be a pandemic, and frankly, we never imagined one of the current proportions, that testing would have to be done on large scale. And I am the CEO of RUCDR Infinite Biologics, which is located on the Rutgers campus and is a contract research organization. Much of our work is for the US government, but also we service all of big pharma. I'll say a bit more about that later. So we decided that we needed to innovate under the pressure of developing a test rapidly. And I'd like to talk to you mainly about practical things, that is the conception, implementation, and management of large-scale diagnostic testing. And to be clear, this test, the development of the test, and this was the first saliva test for SARS-CoV-2, we reasoned from the other coronavirus data that saliva would be a very robust source of SARS-CoV-2. We weren't sure of that until there were several publications in our own work. But Andy Brooks, the chief operating officer, and his team were largely responsible for pushing this boulder uphill and pushing together to get this assay approved. There are now about 80 EUAs, emergency use authorizations, for testing, but ours was the 10th to be approved, and it was approved on April 10th. On May 7th, we received approval for home testing, which was very, very important because prior to that time, the test had to be done in a physician or other health provider's facility. So to say a bit about RUCDR, we're a next generation central laboratory and diagnostic service provider. And these are just typical pictures of RUCDR. As you can see here, sorry, we skipped ahead. As you can see here, we have lots of robots. These are two examples. Now we're capable of doing 50,000 assays a day, and we have 50 of these machines, which are known as KeyMagic 360s. They're a device that can be programmed to extract any nucleic acid from virtually any solution, in this case saliva, although we also do conventional swabs. This does 96 samples at a time, and if you have 50 machines, you can do 50 times 96 samples, which is a lot of samples, obviously. This is a qPCR machine. We had never done any viral diagnostic. We were approved for cancer precision medicine assays, which we had done, but we're no longer doing. We were doing a number of precision medicine assays for big pharma. But again, we had not done a single viral assay. RUCDR is also a biobank with the largest collection in the world of samples from people with psychiatric disorders, alcoholism, and quite a few other diseases. In all, RUCDR has grown in the last 22 years to a very large business, which will hopefully soon be privatized. Some of you may have seen the announcement by Rutgers. So we had all these robots and all these clients. This is just an example. And things dried up because of the pandemic. Clinical trials were suspended. Samples were not coming to us. And we have 200 employees in RUCDR. And I guess one of the motivations to develop the assay, aside from the obvious need, was we wanted to keep all of our employees employed, which I'm sure they appreciated. So in general, we could do this. And I think, and I don't mean to be cruel in saying this, I think anybody on the Rutgers campus could have developed a diagnostic assay for SARS-CoV-2. It's routine molecular biology, reverse transcription, qPCR. At least 24 labs on campus have the equipment and the know-how to do it. Perhaps not, well, for sure, not the equipment to do it at large scale. But I think everybody has to ask themselves the question, why, at a time of extraordinary need in January, February, and March, was that not the case? I think it has to do with the kinds of work we're used to doing. RUCDR is used to handling large numbers of samples. Typically in any one year, we handle 1 million samples. And we're highly automated, and everything is conducted according to an SOP. We have, in a program known as Qualtrics, we have about 600 SOPs which govern everything from how to do qPCR as a recipe to how to clean a microscope and how to decontaminate a sterile hood. So in doing anything, in particular this assay, the science came first. What were the parameters that we had to consider in extracting RNA? How could we stabilize the RNA before extraction so one wouldn't have to immediately analyze either the throat swab or the saliva? And I'll talk about how that was done. The second part was the process. How could we do this in a way where samples would never or couldn't be mixed up, that each sample would receive the exact same treatment. Each sample would be extracted, analyzed, and assayed in the same way. And how do we apply these two principles to automation? So instead of doing anything by hand, we use robots. This is just one kind of robot, a Hamilton. We have many different kinds. Instead of using a routine tube, we use tubes that are designed for automation, for capping, decapping, to fit into 96 well plates. Instead of measuring things by hand, we have automated spectrophotometer, in this case, with a strange name of the company called Lunatic. Instead of looking at the volume in a tube by eye, we look at the volume in a tube by ultrasound. Instead of looking at gels, we use a fluidine system of microfluidics that uses very little material. And this is a slide that I use for other purposes, but it shows that we look at 96 DNA polymorphism SNPs in every one of 1 million samples to determine the gender, the continent of origin, and the individuality. So if there's ever a mix-up in samples, and that can happen, somebody can drop a plate full of tubes, we can actually determine which sample belonged to which person based on the data of these 96 SNPs. So the first question we considered is, what are the best materials to use? And this obviously varies with the assay and disease type. There was a significant amount of data showing that coronaviruses infected the epithelium of the throat, the pharynx, and the lungs. In some cases, it appeared in the lungs first. In most cases, it would appear in the nasopharyngeal canal before it appeared in the lungs. So the question was, what material could we get? Conventionally, as you can see in this slide in the center, sorry about that, one would use a nasopharyngeal swab, which takes a certain amount of skill on the part of the person administering the swab. You can actually damage a person significantly if you're unskilled. And it also means that every healthcare provider taking a swab needs to change their PPE, personal protective equipment, in between each swab. What this means in practice is that a physician, physician assistant, nurse, whatever, can only do a limited number of swabs in an hour. We saw this as a limiting factor to mass testing. So we decided that perhaps saliva, which bathes the throat, and we're not talking about sputum here, which is a different biomaterial, but saliva could be used. If so, the saliva could be collected in a container that we had developed for genetic testing, that it could be adapted for RNA, that one could spit in the tube in the absence of a healthcare professional. It could be done as it was in Middlesex County in the auto inspection facility. It could be done in one's car. It could be done in one's home. It could be done privately in a physician's office. And not only would it not involve a healthcare worker, but it wouldn't involve changing PPE. And if you all remember, masks, gowns, gloves, and such, face shields were in very, very short supply early in the pandemic. And this also was a major issue. So we decided to go full in with Saliva. And there are a couple different kits. We ended up using the one we were most familiar with, the Spectrum kit. I should add, in many of our studies now, we've collected saliva, we've done a throat swab contemporaneously, and also collected blood for antibody studies. Anybody who's in the healthcare workers study, perhaps as a control, as I am, has had this done about a half dozen times. So just to give you an idea of how popular home collection with one or another kits is, this is a market analysis of the number of kits that are distributed each year. And you can see in 2019, more or less, Ancestry.com alone did 9 or 10 million kits. Add that to the 5 million done by, or 6 million done by 23andMe and various other companies, some of which we'd had personal familiarity. It's obvious that these kits were user-friendly and could be done by anybody who took the time to read the instructions. So one of the things I'd like to point out to you at this point, that the way we do research as it relates to the standard of care in medicine is really changing. There are 342 million people in the US, give or take. And in 2019, 126 million hospital visits, 885 million doctor visits, and 13 billion tests were done, diagnostic tests, with 10% for molecular analyses. And this is just a typical genomic test or epigenomic test or transcriptomic test. And now we have various proteomic tests that are coming on board. And even metabolomics is playing an important part. And in many cases, the results of all of these kinds of tests will have to be integrated to determine the nature of a disease that a person might have. So imagine, on average, 5 biosample aliquots for each person who visited a doctor. There might be blood, saliva, throat swab, whatever, urine sample. That would be about 4.4 billion samples. Imagine then the collection of 5 biomaterials each year for 50 years. That's 220 billion samples. The only point I'm trying to make is that in a very short time, the number of molecular samples will exceed the number of conventional tissue paraffin blocks by several orders of magnitude. And in part, this is because the information density in the molecular samples is so much greater, but also they're a whole lot easier to obtain and a whole lot less invasive in general. So, as I said, our assay involved lab automation. This is a gadget made by PerkinElmer. I won't go through it in too much detail. It's just a conglomeration of robots. You have plate feeders here, you have a carousel, and the plates are fed into a key magic, which extracts. Another key magic it's fed into, which extracts. This particular machine, with its automation, plate feeding, and robotics, can do 10,000 DNA, or in this case, RNA extractions a day. We decided these machines were too expensive and they weren't available for assay, so we're just using part of the machine, the Chemagic 360, which is an extraction device which is based on magnetic beads. So we have magnetic beads that bind RNA. There are magnetized spinning rods in the robot to which the magnetic beads will stick. And then the solution that the beads are washed in can allow the RNA to be released from the beads into solution and from there extracted. These gadgets also take up a whole lot of room and are a bit finicky. I should add they cost about $2.5 million each. So one of the keys, and this is what we knew before we started SARS-CoV-2 assay, or I'll just use COVID-19 disease as a shorthand, that sample management was critically important. Samples had to be kept in register during processing and extraction. The register had to be maintained from collection to processing and extraction, and from there to sample storage. We keep an aliquot of every sample so that we can go back and test again if necessary, or if we think of another test that we have subject or patient consent to do. And unless you have this triangle rigid, this is where false negative and positive results come from in laboratories. And in fact, everything that is done in our lab, from the time of collection, every tube has a unique barcode on it, which is recorded at the time of collection. In fact, it's recorded when it's sent to the subject if it's done at home. And the person doing the collection will scan that tube again. That tube goes to the laboratory, usually overnight by FedEx. That barcode is read, and that barcode goes with that sample through every process. And that means if it goes from one tube to the next, that is recorded. And we do this in a very large program in our computer system called the LIMS, a Laboratory Information Management System. And on a single sample, a LIMS will have nearly 100 points of data. Every time that sample moves from one part of the extraction to the other, every time anything is done to that sample, it's recorded in LIMS. And that barcode in LIMS is used for an automated report back to the collection agency. It could be the patient, or it could be a health organization like Vault Health that does a lot of our home collection. Saliva, why is saliva good? Well, we thought it might be a more robust sample, that it might have a higher concentration of virus because it bathes tissues that are infected with virus. We had hoped that we could do home collection without telemedicine. That happened the month after we were approved. It doesn't require, home collection doesn't require patient travel, potential exposure of others. There's a huge reduction in risk to medical personnel, no virus exposure if healthcare workers have done at home. It obviates the need for personal protective equipment, requires far fewer healthcare workers, and large numbers of tests can be done on any one day. which is a bottleneck in many testing paradigms. So we use the Spectrum kit, and here's a simple video of how it works if you haven't done it yourself. And you have a tube with a funnel, you spit in it to a line, you take the funnel off, there's another tube that screws to the top that has a magic sauce that stabilizes the RNA. It's a proprietary solution. We've shown that the RNA is stable for many, many days at various temperatures. There's a bag provided, you put it in FedEx and you send it to the lab. And I'm not going. No, I can't get off the slide. I gotta. Okay. Unfortunately, our test really can't be used. No test available today can be used on people who have neither any symptoms of COVID-19 disease or have not been exposed to an individual who has COVID-19. Although one could assume that everybody has some degree of exposure to somebody with COVID-19 unless they're locked in their house in isolation the full time. So one has to fill out either a screening tool or have a health provider ask questions about whether you have symptoms or might have been exposed. And usually everybody answers yes to the might have been exposed, so we can do the test on virtually anybody. Again, it's a streamlined process. It goes in a bag. The bag is decontaminated overnight to the lab. We're currently using a company called Vault for much of our telehealth visits. You meet with a healthcare professional online who instructs you, just as I instructed you in the video, on how to spit in a tube. If this seems a little ridiculous, it is. I'm sure all of you could manage to spit in a tube all by yourselves. And there are 3 possible results: positive, negative, or inconclusive. Obviously, if you're positive, this is reported back to your health provider and you are instructed to isolate. If it's inconclusive, you're asked to do another test. If you're positive, we like to do a second test. Now, as far as we're concerned, we have no evidence of false positives. And really, the issue of false negatives is when you sample. Viral titers vary tremendously over the course of the disease, and they may in fact be highest maybe 4 to 5 days after exposure or a couple days before the appearance of symptoms. So if we get a negative one day and you're tested the next day, you might be positive just because it was too early to test. So a false negative is really an issue of testing. The way the test is done, we spike every assay with positive and negative controls, and there's no evidence of any false negative or false positives coming from the lab. So what happens in the laboratory? First, we extract RNA from the saliva using the bead method, and that gives us 96 samples every 40 minutes. We have other robots that then move the samples to a 384 well format, which makes it convenient. And at the same time, we transcribe the RNA into stable DNA. We add short DNA probes, oligonucleotides, and perform real-time PCR. and some assays only assay 2. We assay 3 genomic regions in the SARS-CoV-2 genome, a region in the spike gene, in the nucleocapsid gene, in the open reading frame 1AB gene. And we determine the number of cycles, the so-called threshold value, for detection. And if a sample hasn't shown positivity by 37 cycles, it's considered negative. A sample is considered positive if 2 out of the 3 SARS-CoV-2 sequences are amplified, and if the positive and negative controls are correct. So how do you get an emergency use authorization? I think the people in the lab, if I can brag a bit, were exceedingly good at this, considering this is the first time any of them had ever applied to the FDA. We got our EUA on the first shot. 60 subjects were used, and we had independently verified clinical status. That is, they were all sick with COVID-19. This was across 3 hospitals, and we took swabs in universal transfer medium. I find this amusing in that one of the things that kept testing at low level initially was a shortage of universal transfer medium, or viral transfer media, as it's sometimes called. We had a paper that's been published already showing that saline and UTM are equivalent, so one could have used saline at the beginning. At the same time, we collected saliva in the spectrum collection device that you saw a few moments ago, and all samples were processed within 72 hours of collection. At this point in time, we're turning samples around in about 24 hours. And that includes all the communications, which actually take longer than the lab assay. The same volume of universal transfer medium and saliva were inputted for nucleic acid extraction, 300 microliters, excuse me. And the same volume of extraction product, that is 5 microliters, was used in qPCR detection. And I asked that the actual EUA summary be made available, but all 60 samples there were 30 positive and 30 negative. They all tested exactly the same by swab or saliva test. The saliva test usually showed amplification in fewer cycles, indicating that there was a higher titer of virus in the saliva versus the swabs. There was, as I said, 100% positive agreement, and the limit of detection, although that's not a practical limit, was as low as 4 copies per mil. We set the practical limit of detection as 200 copies per mil. And then 100% clinical agreement, 100% concordance between saliva and swab, and all of the assays worked. This data, plus data showing that there was no amplification with 48 common viral and bacterial infective agents, such as other coronaviruses. We had to very carefully choose the SARS-CoV-2 sequence, so it wasn't similar to that of other coronaviruses. There was no cross-reactivity. So not only is the test sensitive down to about 200 copies of the virus per 200 mil, but it's specific. There was no cross-reactivity with 48 other organisms. And as I said, a month later, the FDA approved saliva collection at home. And this is actually a picture of that big robot that you saw a while ago in schematic. What do you need to do a test? That's what probably was so intimidating to people that they never thought to devise a test in their labs. I suspect most labs could have devised a test within about 2 weeks. We received EUA approval on April 10th for collection. By May 10th, we had the formal approval for home collection. Now, EUAs are peculiar. This EUA is only for assay in the Rutgers diagnostic labs. So you couldn't take the recipe for our test, put it in your own lab, and be EUA approved. Also, as is the terms of any diagnostic test that's FDA or EUA in this case, which is simpler, approved, there are only certain devices that will suffice, and they have to be described in the EUA. So we have the kinds of swabs, the kinds of collection device, and this is very key. The automated extraction chemistry, we use a commercial COVID-19 detection kit that we developed with Thermo Fisher, PerkinElmer nucleic acid detection kit, and either QuantStudio or via 7 quantitative cyclers. Also, to do it in New Jersey, you have to be New Jersey Department of Health approval, and then we received such approval from New York right afterward. If you receive New York approval, you can pretty much do the assay in any state because they accept New York approval. So testing was initiated in early May across hospitals, telehealth, healthcare networks. About 3 weeks earlier, we were testing in various counties. We were testing in nursing homes. We were testing first responders. And first responders, people in facilities, were our highest priority. This is what FDA approval looks like. It's just a letter. This is the document that you all have. And as you can see, it's very specific: nasopharyngeal swab, anterior nasal swab, turbinate, etc. And it's limited to the Rutgers Clinical Genomics Lab at RUCDR. And one of the things that happened, there was an initial delay in testing because it wasn't clear how we could get paid for this. One of the things that I was forced to do as CEO is borrow the money to get the testing in place at large scale. So I actually had to borrow about $7 million to make this all happen. And it would have been nice to pay that money back. And to do that, we would require compensation for each test. But the CARES Act, which was passed by our Congress, requires payer coverage for testing. So if you have insurance, your payer will pay for testing. If you don't have insurance, the feds will now pay $100 per test. And that's the end of my talk. I'd be happy to answer any questions, and I'll show you.
Thank you very much, Jay. We have time for a few questions for Jay before we close. Please use the chat box, or looking for raised hands in a participant list approaching 100 people is a bit challenging. So you got a thumbs up, another thumbs up.
Thank you.
Any other? So why don't you take us off screen sharing, Jay? That's your control at the top of the screen. Top of the screen to the right of the.
Yeah, I see it. I see it.
A little bit of red. Yeah. Okay, great. Thank you. Yeah. What does cross-reactivity refer to? A question from Luis Alfaro.
Cross-reactivity refers to a positive result that is an amplification of a gene sequence in another organism. So if, for example, the SARS, the original SARS virus, cross-reacted in our PCR reaction, we could detect it or any of the coronaviruses that cause colds, but our primers don't bind to those viruses and don't amplify the sequence. So we don't see a positive result from those other organisms. There are many, 48.
Other questions for Jay?
I can't see you all, so I.
Yeah, no, it's Zoom brings with it a different set of challenges.
Well,
One
thank
new.
you.
Hold on a second. Okay, that was the sorry. That flag was for the question that you've just answered. So, what's your current capacity, Jay?
We can do. We've taken over space in the Waxman, the newly built facility, and I think that's almost in place. Those people in the Waxman saw 30 of those robots delivered. Their jaws kind of dropped. It should be ready very soon. We'll be up to 50,000 samples a day, or about 300,000 a week, which really only scratches the surface of what we need in New Jersey.
Absolutely, as far as New Jersey is concerned, but for contact tracing testing within the Rutgers community, you've got that covered.
Yeah, we have 10,000 tests a week dedicated to Rutgers. We're currently the largest testing facility in the state.
Yeah,
yeah. I have a quick question. Jay, what do you think about pooled testing? I've read in places that might help test a larger number of people.
Could you repeat that? What kind of specificity?
Pooled testing, pooling multiple samples.
Yes. I'm sorry. I've looked into pool testing extensively. I mean, theoretically, if you pool 10 samples, the pool comes back negative, you're great. If the pool comes back positive, then you can test each of the 10. The number of samples you would have in a pool would depend on the fraction of infected people in the community. And there are newly designed algorithms to come up with optimum pool sizes for different levels. Several problems, although the FDA just a couple days ago indicated that they were willing to consider pool testing. Right now, pool testing cannot be done for clinical samples. So it would have to be done on an IRB approval as research, which I think is perfectly reasonable. And then if you have a positive pool, you test everybody clinically. I'm not sure that the PCR method is best. For example, if you did the cDNA synthesis with barcodes, you could theoretically take several hundred people, do deep sequencing, and see if you detect any of the SARS-CoV genome. So I think there are other methods. There are also some very nice isothermal amplification methods, which make the test a little more applicable to less automated labs than ours.
So there are a few more questions, Jay. Let me just hit you with them. So question from Latricia. If someone has taken the test, receives a negative result, could that result be used for travel to other places requiring negative results?
That's a legal issue and a governmental issue. As far as I'm concerned, yes, I don't let my children and my new grandson into the house without testing.
Fair enough. Dr. Jiang mentions that he or she has done the test and very much enjoyed hearing about what's under the hood. Erica
Thank you.
asks if the price of the test will go down as you test more people.
Well, depending on who does it, you have different costs involved. So if a test involves Vault Health, they have to pay for, that's not part of Rutgers, I should add, or RUCDR. They have to pay for their infrastructure. They have to pay for the person who does the telemedicine visit and everything else. Right now, we're using a company called Accurate Diagnostic as the clinical intermediary because RUCDR is not a clinical laboratory in the sense we're not a healthcare provider. So the healthcare provider has to order the test and give it to us, and they take a piece of the price. But yes, if we eliminate the healthcare provider, something they're not looking forward to, the price of the test could go down. Our break-even point is below $100.
Okay, well, maybe we don't want the people you're negotiating with to know that.
I think they do. I mean, RUCDR is entitled to make a reasonable profit to move forward and make more investment in its infrastructure.
Yeah. So Melanie Ortiz Alvarez, who's a resident of Puerto Rico, asks, what's your opinion of the efficacy of serological testing?
Oh, that's the most common question I'm asked. It's a very good question. Serological testing is entirely dependent on the antibody assay that's used and how it's done, whether it's done by the more complicated laboratory ELISA or one of these simple tests that looks like a pregnancy test. You can certainly have had the virus. There's ample data, especially in children and older people. You can have had the virus and not develop an immune response. Sure. So that's absolutely clear. 2 articles appeared this week in Lancet on that particular point. But most people will develop an antibody response in 14 days. It's IgM, IgG, the order in which they occur might be different in different people. And then the question becomes, how long does this last? It may only be a few months in some people. People have had mild disease develop much lower titers. And then the question becomes, what level of antibody titer protects somebody from secondary, from reinfection? So I think one has to interpret the results of serological testing very, very cautiously. And we don't know to what extent, as was alluded to, I think, in the second talk, that exposure to other SARS viruses might cross-react to SARS-CoV-2, that past exposure. We don't know.
And then we'll take this as the last question since we're coming up on 3, which takes us half an hour over our original time.
Sorry.
You don't need to apologize. Are saliva testing procedures and methods being shared on an international level?
Yes. As all of you can see, I trust it's been made available. I asked that. Our EUA is posted online at the government EUA site, and anybody can get it. And for very modest costs, a few $1,000s, Rutgers, and it's not me, it's the Office of Technology Transfer, will share the detailed SOPs with whoever wants them. But again, they would have to get their own EUA, and they'd have to prove to the FDA that they can do the assay as carefully as we do it.
Absolutely. So with that cautionary and important comment, I need to draw this to a close.
Thank you all.
Well, thank you, and I want to thank Steen Hoyer, Sarah Carey, and you, Jay Tischfield, again for terrific contributions to a very interesting afternoon. Thank you very much.
Thank you, Stephen, for your contribution as well.
Well,
for leading the effort.
It's an honor to be able to talk about the PDB.